purposes, such as the COVID-19 vaccine. So there are challenges, though, to get the mRNA. So if we want to deliver CFTR mRNA, or maybe do CRISPR Cas9 kind of CRISPR mRNA, kind of gene editing mRNA delivery, there are challenges. And so in this cartoon here, one of the things is there's those extracellular barriers. So how you could actually get the mRNA degraded, or you could actually get immune response, even though you can actually make modifications to mRNA to prevent this, and you can get degradations. So as a result, there's been efforts to try, can you encapsulate and protect that mRNA for a more effective delivery. Now, there are these, however, if you want to deliver it, the challenges with cystic fibrosis, you have these extracellular barriers. So one of the things I mentioned is you have thick mucus. As we see here in the EM on the left in normal, on the right, you have healthy mucus. As the laser pointers show, you have this pore size, you have these nice pores, you're gonna get clearance by the cilia, as shown left here. Now, with patients with with cystic fibrosis, you see this thickened mucus as seen here on the left-hand side. And so you have a decrease in the pore size, and also in the bottom cartoon, what you see is the mucus with people with cystic fibrosis is more viscoelastic. There's change in pH. There's changes in kind of in the ionic strength, and there's changes, and it's heavily charged because of the mucus is very kind of negatively charged, and because you have this barrier which prevents really a lot of drugs, almost up to 99% of small molecule drugs are unable to penetrate through this barrier. And so if you now want to develop delivery systems, how do you do that? Well, there's been efforts by others and ourselves to try and use polyethylene glycol, or PEG, to densely coat the surface of drug gene carriers to then deliver across. So what happens, what I want to show here is the PEG on its surface essentially it's a muco-inert coating. It allows for diffusion through the kind of the mucus barrier. And on the upper left, what we have is if you have a particle that doesn't have a PEG coating and you deliver it to the airways, you actually get poor distribution kind of in the bronchioles, in the airway space, and you get aggregation. Now, if you coat it with PEG, these carriers, you'll get more uniform distribution, as we see on the fluorescence micrograph on the bottom. You get more uniform distribution. And then on the right, what this is effectively showing is on the bottom right, as you, if you coat your drug gene carriers with PEG, you get better diffusion through the hydrated pores of your CF barrier, your mucus barrier. Whereas with uncoated, up on the upper left, uncoated particles, what we see is the diffusivity can be 1,000-fold less. And so this is the advantage of using this PEG coating. And we have shown this also. So don't want to belabor the point. Now, if we come back, we have that extracellular barrier, the mucus, but then the challenge is, well, how are we going to deliver that mRNA? And we talk about that using kind of carriers to encapsulate that mRNA to get it through the extracellular barrier and then intracellular to the, into the cell, so then it can, get into the endosome and then have effective endosomal escape and release into the cytosol for then protein production. So there have been different carriers that have been used for cystic fibrosis. A lot of early efforts have been focusing on viral vectors on the upper left, such as adenovirus and AAV. While they have their advantages, viruses are naturally evolved to infect cells, especially even our airway epithelia. However, there are challenges with, say, immunogenicity. And in the case with these viral vectors, you can have packaging constraints. So with AAV, it can only package about 4.7 kb, so it can limit how much of the CFTR gene can be packaged. But it's still very promising. But there are efforts to kind of look at non-viral vectors. They offer the advantage of reduced immunogenicity. You don't have the issue of packaging constraints, and you can deliver various types of cargoes of different sizes, such as plasmid DNA, your mRNA, or even, like I said, kind of your CRISPR Cas, your gene editing components. So you don't have those size constraints. And so we're very much interested in sizes such as plasmid DNA, your mRNA, or even, like I said, kind of your CRISPR Cas, your gene editing components. So you don't have those size constraints. And so we're very much interested in the lipid nanoparticles, as many of us are. And lipid nanoparticles, there's been your gene editing components. So you don't have those size constraints. And so we're very much interested in the lipid nanoparticles, as many of us are. And lipid nanoparticles, there's been extensive document literature about its promise to encapsulate, nucleic acids, as with kind of the various COVID-19 vaccines and various, actually in the CF field, there's been work, to encapsulate nucleic acids, as with kind of the various COVID-19 vaccines and various, actually in the CF field, there's been work trying to use kind of lipoplexes and liposomes to try and deliver, and they've entered clinical trials. However, there are still challenges to improve their, really for airway delivery, to improve the functional output. And so that has been a challenge, but there's been ongoing work. So there's, of course, with lipid nanoparticles, they're kind of non-toxic, and they're very well characterized. So very much interested. However, one of the challenges with using non-viral systems, well, any carrier system, is the effect of aerosolization. As I mentioned before, while CF is a systemic disease, most efforts are trying to improve, kind of restore the airway function so you can breathe. And so one of the challenges with using these carriers is eventually you'll have to put it into, you'll either want to put it in nebulized form, so you want to aerosolize it, or put it in a dry powder form where then you can then inhale it. And then once it inhales, it still has to penetrate all the way through, get to the bottom parts of your kind of your airways where they can penetrate, as we see here, the mucus, excuse me, go through cilia and get into the affected airway epithelia that has these affected mutations. Now, one of the challenges with aerosolization, you have the shear stresses that, due to aerosolization, that your carrier is subject to, and even your nucleic acid. So that's something we need to think about before we, to consider. So this gets at our ultimate sort of question is, there's so many barriers. The aerosolization aspect, getting through the mucus barrier, and then, excuse me, the intracellular barrier. Can we develop a way to kind of do a high throughput screen where we can screen different lipid nanoparticles that encapsulate mRNA that are still stable enough to then achieve effective mRNA delivery in cell lines, but also in in vivo, in animals? So our general strategy was to use a design of experiments approach. And the idea here is we would develop a library of different formulations, of different lipid nanoparticle formulations. And so in the cartoon, as I'm circling, in our lipid nanoparticles, we're using sort of kind of the very canonical components. So we have our cationic ionizable lipid that we use. We have our PEG lipid to provide, in this case, for pulmonary delivery, to get that mucus penetration. We have our helper lipid, our helper phospholipid, and excuse me, we also have our one moment. We have our structural lipid, our cholesterol, and so we have these four components along with our mRNA, and the idea is, can we combine them at, make different formulations of different amounts, ratio, and then look at how effective they are. So we want to make this library. The reason why we want to use a DOE, a design of experiments approach, is we, wanted to really look at all of our parameters as a whole, as opposed to just kind of changing one factor at a time, as opposed to changing, say, modulate just the our ionizable cationic lipid. Can we look at all the parameters at once? And after that, we would characterize their properties. After formulating it, we would characterize their properties, see how nebulization or aerosolization affects those properties, test it on cell lines, and then see if we have any lead candidates see how they'll work in vivo. So we came up with, based on the literature, with lipid nanoparticles delivering siRNA and mRNA, we came up with our components, and we came up with sort of a lower limit and an upper limit. And so these are sort of all the components and kind of the range that we're looking at in terms of our molar ratio. So the contribution of each component to the formulation. And so this was pretty much these values, these lower and upper limits were taken from the existing literature of what's been out there, what's been published. And so on the bottom is sort of the various compositions of different phospholipids we tried, or helper lipid, the different PEG lipids that we tried, and then those compositions. And so we kept, we just used basic cholesterol, and we actually used the MC3 ionizable lipid that's been very heavily, used out there. For those who aren't aware, we actually use the Precision Nanosystems Nanoassembler to achieve high throughput rapid assembly of these formulations. So we made 18 formulations, and this allowed for very rapid mixing where we could take our lipid components in the ethanol phase, our organic solvent, and then our mRNA in our aqueous solvent with the rapid mixing make our particles. And so this allows for kind of rapid assembly of our formulations, which allows for very easy testing downstream. And since we're using the assembler, this allowed us to actually achieve quantities that were sufficient for not only cell testing, but also animal work, as I'll show later. Okay, so before we did the DOE, what we kind of tested the different formulations, we wanted to figure out first, what is the fix? We wanted to fix our N to P ratio. This is essentially the amount of amines to phosphates, and really what's affecting the number of really our particles, our lipid nanoparticles, to the ratio of phosphates due to the mRNA. So that N to P ratio. We want to figure out what is the optimal ratio. We want to keep that number fixed before we start changing all the other components in our DOE. So what we did is we actually using Alnylam's Onpattro formulation. We used all their components and we just changed the ratio. Instead of siRNA, we used mRNA, and we changed the molar ratio of our N to P ratio of our lipid nanoparticle. So we went from 6:1 to 200:1 ratio. And what we did is we made different formulations, mimicking the composition of the Onpattro formulation. And we delivered a GFP reporter, mRNA reporter, and we looked at cell, essentially expression of GFP through flow cytometry. And what really the